Hi friends, welcome back to my channel. In this video, we are going to talk about an important experiment which proved that the DNA replicates semi-conservatively. I will explain what does that mean by Matthew Meselson and Franklin Stahl in 1958. Please make sure to like, share and subscribe my channel. So, in 1958, Matthew Meselson and Franklin Stahl they performed an important experiment which actually proved that DNA replicates semi-conservatively, which was proposed by Watson and Crick. So, what the semi-conservatively means is that when it replicates, so in DNA there are two strands of DNA, and semi-conservative means half. Like during replication, the one strand remains the same, and then there is formation of the new strand from the one old strand. So, DNA replications is the mean to produce new molecules that have the same base sequence, and it occurs during the interface of the cell cycle, especially in the S phase. So here you can see the cell division. It starts with the preparation for the DNA synthesis from G1 phase or sometime from G0 to G1 phase, and then it moves to the DNA replication phase, which happens mostly in the S phase because it's called the synthesis phase. So during this phase there are always two strands of the DNA, and then it forms new strand by like the two strands they get separated, and then each of the strand act as a template to make new strands. So how does this happen? So there were three models of DNA replications that were suggested during that time when nobody knew how DNA replications happen, like what is the way. So they created this way, like a daughter duplex. It's semi-conservative, means the duplex that we have, the one is made of the parental strand and the other one is newly synthesized. So this could be one way. The other is called conservative, in which the two strands that are present, they serve as a template for new strand, but in the end they stay together. So we have one like DNA duplex of the original strand and one DNA duplex of new strand. So in this way the original strand remain conserved. And there's dispersive, which means the random, like there is mixing of both the duplex, like the new and old strand, in the parental and the new daughter strand. So it's like this. So what we have talked about is conservative. They divide and they produce from one strand to the two strand, but in this case the original strand remains the same and there is formation of new one, and from this original it again form the new and the original one. So in this case always we will have the one original strand and then the new strand. So this is called conservative. The another one is semi-conservative, in which one strand is from the parent, as you can see here, and the other strand is formed new. So in this way you will always have the new strand, the old strand always. And the another one is dispersive, in which always there is mixing in one strand of both the new strand and the old strand. So uh, what method is used for DNA replication at that time they didn't know. So they did a very cool experiment which was the called Meselson and Stahl experiment. So they begin by growing the E. coli in a media which contain heavy isotope of nitrogen. So because DNA is made up of nitrogen, like it contains the DNA structure that you have, the basis that they contains nitrogen. So they use an heavy isotope for nitrogen. So it's 14, but they used a heavy one for the original strand 15, and because then it will be easy to detect which nitrogen it contains. And then because as I told you that bacteria took up nitrogen to generate more new biological molecules include DNA, and after some generations they shifted the bacteria to like after few generations they shifted the the new media nutrient with the normal nitrogen, which is called the light 14 nitrogen isotope, and allow them to grow several generations. and then they separated them based on the cesium chloride centrifugation. So in the cesium chloride centrifugation, the lower one will um, so there is separation because of the weight, right? Because the heavier one will be separately distributed, because this is a density gradient. The heavier the isotope, the more density they will have. So based on the density, they can be separated very easily. So if the replication is by conservative means, so this will be completely 15-15, right? Because both the original strand will contain 15 nitrogen, the heavy one. And when they do the replication, so this should be 15-15 and this should be 14-14, the new strand. And then we will always see either 15-15 nitrogen or 14-14, but never the mix. If it is semi-conservative, we will see that the original strand contains the heavy nitrogen, but the new strand contains the light nitrogen. So the density will be the intermediate of 15 and 14 or 14-14, but never 15-15 in the daughter strands. And if it's dispersive, then always it will be the mix. It will never be pure as conservative or semi-conservative. Like we will never see nitrogen 14-14. We will always see the mix of both. So like we have talked about, they have the original strand, like which contain the nitrogen, heavy nitrogen, and because it is heavy, it will sediment faster, so you will see it at here. And then they've shifted the media to the 14 nitrogen. So now if, like we have talked about, if it is conservative, right, if it is conservative, we will see either 15-15 or 14-14. So in this case, you can see we saw the intermixed of 14 and 15, right, which can be, so if it's 14 and 15, it can be either these ones or this ones, right? Because in conservative, we will always see the 15-15 as well, but only in semi-conservative and dispersive we will see the mix, and this is what we saw. So now we excluded that it's not conservative. It can be either semi-conservative or it can be dispersive. And then after some generation, if it is semi-conservative, we will see 15-14 and 14-14, never 15-15. But in dispersive, we will always see 15 and 14, never only 14-14. So then what we saw is 15-14, 14-14. So it was a mix of the hybrid one, which is the intermediate of heavy and light, and the light one, which is the case for semi-conservative. So by this experiment, they proved that the DNA replicates semi-conservatively, which means that both the strand get separate, both of them act as a template for the new strand and then generate daughter strand. So I hope it's clear. If you have any doubts, please let me know in the comment section. And in this way, they prove that DNA replicates semi-conservatively. And please make sure to like, share and subscribe my channel. Thank you.